Bacterial cell structure. So identifying about the organelles or different types of structures or functional units which are present in the bacteria will be very essential to understand in detail about bacteria. So my name is NH Shankar Reddy and I am doing plant pathology in Annamalai University, PhD plant pathology. And we'll see what are all the organelles are different types of structures which are present in the prokaryotic cell, that is bacteria. You'll see here different types of structures, that is plasmid, pili, ribosomes, nucleoid or nucleus, endospore, flagellum, cell wall, plasma membrane, mesosome, outer membrane, and inclusion body. These are all the important and different types of structures or the functional organelles which are present in the bacteria. So here we will see one by one. So surface appendages. Though coming to here the components which are divided based on their based on their presence, or so here there are some divisions, that is surface appendages. So flagella, pili or Fimbriae, that are comes under surface appendages. Then cell envelope, so capsule or slimy layer or cytoplasmic membrane or cell wall, these are all comes under cell envelope. Then cytoplasmic organelles, chromosome, plasmid, ribosomes, mesosomes, storage granules, and other inclusions are comes under cytoplasmic organelles. And specialized structures, that is endospores. Endospores which is produced by some few specialized bacteria, especially gram-positive bacteria. So now we will see one by one. So the first one is flagella. So here on the right side diagram we can see here, this is flagella. So this is flagella. So the main function of flagella in bacteria is a locomotion. So the flagella is the plays a key role in locomotion of the bacteria. So which arises from the cytoplasmic membrane, where this flagellum arises from the cytoplasmic membrane and form a granule called blepharoblast. We can again we can say otherwise, like where the blepharoblast gives the stability in cytoplasmic membrane where the flagellum can arises. So this blepharoplast can be the stability point, like that, holding point like that, which is arises from the cytoplasmic membrane. We will see few of the important things about flagella. Then it is a filamentous structure which are attached in the cell surface, mainly helpful in the locomotion of plant pathogenic bacteria. Here flagella consists of a protein called flagellin, where this flagella contains a protein called flagellin. So it is actually very small. Normally this flagellum usually ranges from normally a few hundred times longer or larger, sorry, not larger, longer than bacteria. So it can't able to seen in the light microscope unless we can stain it, because of a small thin structure we can't able to stay with the normal microscope. So we have to stain it to see the flagellum. It is a locomotory organ which is a very thin thread-like structure and arises from cytoplasmic membrane. It contains a protein called flagellin. Now we will see the next. So different types of flagella arrangements. Now we will see what are all the bacteria which produce the types of different types of flagella. Now we will see one by one. So this table will be very important, because these questions can be repeated, asked in different types of exams, that may be in JRF or SRF or national eligibility test, whatever it is. So to understanding about bacteria which produce different types of flagella will be very essential and crucial. So now we will start with the atrachus. Atrachus means no flagella or flagella are absent. Here we can see the diagram on the right side. There is no flagella present here. The example is xylella. Then monotrichous or polar flagellum. Here monotrichous means only one flagella is present at one end of the bacterium. One flagella is present at one end of the bacterium is called monotrichous. That is example Xanthomonas. Then amphitrichous. Amphitrichous means a tufts of flagella or a single flagellum which is present on both ends, not single at both ends. A tufts of flagella or single flagellum which is produced on both ends is called peritrichous. Example Pseudomonas species. Then peritrichous. Peritrichous means, peri means many or plenty. Here peritrichous, the a multiple number of flagella or flagella all over the body. The flagella can be arised from all over the body. That is the example of peritrichus. Example is Irvinia. Then Lopotrichus. Then Lophotrichus. Lophotrichus means a tufts of flagella which are present on both poles. Which are present on both poles. This is the example of Lophotrichus. A tufts of flagella which is present on both poles. That is called Lopotrichus. Here Cephalotrichus. Cephalo means a group of flagella which is present on one end. Here actually here they are wrongly placed. It is a Lopotrichus. It is not Lopotrichus. Cephalo means head. In head only one side only we have space to arise flagella. You can remember that cephalo means head. Trichus means cephalotrichus means a group of flagella which are arised from only one end. That is called cephalotrichus. Actually it is wrong. It is wrongly placed here. Cephalo means a group of flagella which are arranged from only one end is called cephalotrichus. Example Pseudomonas fluorescens. And the next one is subpolar flagellum. Subpolar flagellum, which means the bacteria with a lateral flagellum at only one side or sometimes both sides can be arranged. Best example is Agrobacterium. So these are all the different types of flagella. Now we will see pili or Fimbriae. So there is a lot of difference between flagella and pili or Fimbriae. I will tell you in a single word. Whereas flagella is the locomotory organ of bacteria, whereas pili is the sexual organ which is helpful for conjugation. This is the organ which is helpful for colonization of bacteria, that is pili or Fimbriae, otherwise called as Fimbriae. Now we will get into it. We can, in the right side we can see the diagram is a pilus. It is a pilus or Fimbriae. Here it is a air-like appendages which is usually shorter, straighter and thinner than flagella. Now usually flagella is thinner. It is thinner than flagella, that we have to observe this, and it consists of a protein called pilin, whereas flagella consists of a protein called flagellin, but pili consists of a protein called pilin. Pili consists of a protein called pilin. Now we'll see what is the major function of pili. Here the major function of pili that I already told you, which determine the virulence that allow the pathogen to adhere with the host surface for colonization. Simply conjugation. On the right side we can see here that is the donor bacteria and this is the recipient bacteria. It produces a long tube-like structure here. This long tube-like structure is called sex pili or Fimbriae, sometimes otherwise called as Fimbriae. This pili is the simply acts as a conjugation bridge. So here the function of sex pili is a donor is from where a DNA of a donor is transferred from donor cells to recipient cells. The main function of pili is it allows the DNA to pass over from the donor bacterium to recipient bacterium. That is the main function. Here the last one is it is helpful for bacterial conjugation, resulting in the transfer of DNA from one cell to another cell, from donor cell to recipient. That is the main function of pili. The main difference between flagella and pili is flagella is the locomotory organ, whereas pili is the conjugation organ. Or simply what can we say that the organ that can that can create genetic variations are helpful for colonization. Yes. So now we will see the next one. Then capsule or slimy layer. Capsule or slimy layer. Here we can see the capsule here. The different type of structures I already told you there in capsule. Now we will see in detail about capsule. So whereas the capsule is made up of polypeptides and proteins. Polypeptides and proteins. It covering is loose masses. When it covers with the loose masses, it is called slime or slime layer. When it is a well defined or when it is a clearly defined, then it is called capsule. Then it clearly defined, it is called capsule. So depending on the thickness, here the capsule can be divided into two, that is macrocapsule and microcapsule. So macrocapsule, the size ranges from more than 0.2 micrometer in diameter, whereas microcapsule less than 0.2 micrometer in diameter. What will this capsule or slimy layer functions as? It mediates the attachment of bacteria to the host surface. I already told you that slime or slimy layer, it's like little bit slimy, so it can mediate the bacteria, that can helpful for the attachment of bacteria with the host surface, so in few of the times it also functions as it to protect the bacterium from phagocytosis with blood cells or predatory protozoa. Whatever, not only predatory protozoa or phagocytosis of blood cells, this slimy layer will also help to protect the bacterium from other bacteria that may be produced by the toxic chemicals or whatever it is. And the one more very important function is this capsule protect the bacteria against drying or desiccation, because if the temperature or environmental conditions changes outside, what will happen? Definitely there will be a desiccation, or extracellular metabolic activities will happen. So what will happen? This capsule will protect the bacterium against drying or desiccation. That is the major and function of capsule or slime layer. So now we will get into the plasma membrane. It is also a lipoprotein layer which maintain the cell turgor. So exchange of molecules, different types of fluid, that may be carbohydrates or glycoproteins or the internal proteins, whatever. It can maintain the cell turgor, exchange of different types of material inside and outside. It can maintain cell turgor. So it also regulate the transport of material entering and exiting the cell. I already told all the materials which are entering into the bacterial cell, which are going out, entirely it is regulated by plasma membrane only. Plasma membrane mainly functions to maintain the cell turgor pressure, and it also helpful to regulate the transport of materials that is entering and exiting into the bacterial cell. Then the next major structure is cell wall. So here uh, bacterial cell wall is normally made up of mucopeptides or peptidoglycans, whereas already initial classes I told you that a fungal cell wall is made up of chitin here, but bacterial cell wall is made up of mucopeptides or peptidoglycans. It is very important. So these mucopeptides or peptidoglycans is a polymer of N-acetyl glucosamine. Remember, N-acetyl glucosamine and N-acetyl muramic acid, NAG and NAM, constitute to form beta-1,4 glycosidic linkage. Here the bacterial cell wall is made up of mucopeptides or peptidoglycans. Remember, peptidoglycans are mucopeptides. So this peptidoglycans or mucopeptide is a polymer of NAG, N-acetyl glucosamine, and NAM, N-acetyl muramic acid, constitute to form beta-1,4 glycosidic linkage. This is where the bacterial cell wall is made up of. So it will be very important. So there is a chance to ask in different types of exams. Here that has mentioned the composition of NAG and NAM. This is the composition of NAG and NAM. So now we will see. So bacteria, based on the cell wall or rigidity or different types of layer, the bacterial cell wall can be divided into two, that is gram-positive bacteria and gram-negative bacteria. Now we will see the difference between gram-positive and gram-negative bacteria. What are all the differences the gram-positive and gram-negative bacteria having? Starts with cell wall. Here the gram-positive cell wall is made up of 85% of mucopeptides, whereas gram-negative cell wall is made up of just 3 to 12%. But the remaining composition is composed of lipoproteins and lipopolysaccharides. That is the first main difference between gram-positive and gram-negative bacteria. The gram-positive bacteria cell wall contains major amount of mucopeptides or peptidoglycans, that is 85%, whereas gram-negative bacteria cell wall is made up of also mucopeptides, but it contains very less amount compared to gram-positive. There is just 3 to 12%, but the remaining composition is made up of lipoproteins and lipopolysaccharides. And the presence or absence of teichoic acid or lipoteichoic acid. The lipoteichoic acid is present in gram-positive bacteria, whereas the same lipoteichoic acid or teichoic acid is absent in gram-negative bacteria. Coming to rings, the gram-positive bacteria contains two rings only. Two rings, these are present on flagellum that support the rings. The flagella has these two rings. Actually, this flagella contains these two rings which supports. the peptidoglycan layer and plasma membrane layer, arises in between. So whereas this M and S rings arise in between peptidoglycan layer and plasma membrane layer, the two rings are M ring and S ring. So gram positive bacteria contains two rings, M ring and S ring, whereas gram negative bacteria contains four rings, that is L ring, P, M, S. So the two rings is contained by positive bacteria, whereas four rings is contained by negative bacteria. Here one more important thing is staining. During staining, the gram positive bacteria will produce violet color or purple color, purple color, whereas gram negative bacteria produce pink or red color. This is the identification factor for gram positive and gram negative bacteria while staining. Remember, violet or purple color is produced by gram positive, whereas pink or red color is produced by gram negative. So here the sensitivity of antibiotics. So very sensitive to see, both this gram positive and gram negative bacteria are sensitive to penicillin, resistant to penicillin, but sensitive to tetracycline. Here the cell wall is made up of cell wall type, in gram positive bacteria the cell wall type is protoplast means completely digested cell wall or completely lack of cell wall, which is bounded by the plasma membrane. So that is completely digested cell wall is called protoplast. So this type of cell wall is contained by gram positive bacteria, whereas partially damaged cell wall or not completely removed cell wall, that is called spheroplast, which means partially damaged cell wall or not completely removed cell wall, can be seen in gram negative bacteria. The cell wall type in gram positive is protoplast, whereas the spiroplast is the gram negative bacteria cell wall type feature. So the coming to the example, Bacillus, Streptomyces. Let me tell you an easy remembrance. Where we are doing this entire course is because only for B.Sc students, Bachelor of Science students, Agriculture. You remember in gram positive, B.Sc, B stands for Bacillus, S stands for Streptomyces, C stands for Clostridium or Corynebacteria. So in gram positive, B.Sc, you remember B.Sc, you're studying B.Sc, so you remember B.Sc, B stands for Bacillus, S stands for Streptomyces, C stands for Clostridium. Whereas in gram negative, most of the plant pathogenic bacteria are gram negative. I will give a few of the examples, that may be Pseudomonas, Xanthomonas and Salmonella. So remember, gram positive means B.Sc. You are studying B.Sc agriculture, so B.Sc gram positive. B stands for Bacillus, S stands for Streptomyces, C stands for Clostridium or Corynebacterium, whereas the remaining all plant pathogenic bacteria comes under gram negative only. Now we will see the chromosome or genome. It's a very important in bacteria. On the right side we can see the diagram. This is the genome. This was extended or enlarged form of bacterial genome or DNA. It is the genetic material of bacteria which is bounded in a histone protein. The thing is, this chromosome not contains any nuclear membrane. It contains no nuclear membrane can be observed. Here all the genetic information, whatever the bacteria will require, it contains in the chromosome. Usually this chromosome is several hundred times longer than the length of the bacteria. Mostly it ranges from 1000 micrometer in diameter. In the introduction of bacteria itself, that I told you, the size of bacteria ranges from 1 to 2 into 2 to 5 micrometer in diameter. Just 2 to 5 micrometer in diameter. If you see this chromosome, the length itself will be 1000 micrometer in diameter, which means several hundred times longer than that of bacterial cell length. Here the bacterial chromosome is called Genophore. So all the necessary information can be stored in this chromosome. Here the important thing is, the bacteria contains only one chromosome, or one DNA. It doesn't contain plenty or multiple amounts of DNA. It contains only one DNA. Now we will see the plasmid. So definition of plasmid is, it is you can see here plasmid. It's not clearly defined here, but let me tell you. So a small covalently closed circular extrachromosomal DNA segment. You can hear small covalently closed circular extra chromosomal DNA segment is called plasmid. We can see here plasmid is small, closed, covalently closed, circular in shape, double-stranded. Remember, double-stranded DNA segment is not single-stranded, double-stranded DNA segment which have their own capability to replicate individually. It is the very important. Actually, in here all the genetic information which is present in chromosome, that is the genophore only. So rather than that genophore, it also contains some genetic information. It has a capability to replicate them individually. This is a very important factor that we have to consider here. Then plasmids are also known as, there is some other names of plasmid, that is sex factors or conjugates or extra chromosomal replicants or transfer factors. These are all the different types of names that plasmid having. Remember, it is a small, circular, covalently closed, extra chromosomal, double-stranded DNA, not single-stranded. Remember, it is a double-stranded DNA. Already told you it contains extra information. What will be the function of this plasmid? Coming to get into this, it contains 1 to 5% of extra information of genome or genetic information that will be helpful to protect the bacteria against antibiotic resistance. The first function is antibiotic resistance and tolerance to toxic chemicals or production of toxins or synthesis of enzymes. These all are functions, or if another bacteria are producing some maybe antibiotic chemicals or the toxins or enzymes, this information or the amount of information that is present in the plasmid help to protect the bacteria against all these toxic related things. Now we'll get into the ribosomes. Ribosomes, whatever, wherever the ribosomes are, the main function is protein synthesis. But the type of ribosomes which is present in the bacteria is 70S type of ribosomes, which are normally present in the bacteria, which majorly functions as protein synthesis. Protein synthesis. The next one is mesosomes. Mesosomes, so these are all a large investigations of the plasma membrane which are irregular in shape. Now we will see the few of the functions of mesosomes. These are the site of energy metabolism and respiration. Mesosomes, site of energy metabolism and respiration. It is also the site of attachment of DNA membrane, which plays a major role in in binary fission. What will happen? This mesosome, sometimes this small septum will enlarges and helpful for the bacteria in binary fission times. It is also the site of energy metabolism and respiration, and the small finger like constrictions which are present. Sorry, I forgot to place the diagram, but the small finger like constrictions would help to form the septa during binary fission. So during binary fission, the small septa will helpful to form the septum, so that will be helpful to divide the bacteria equally. Some exoenzyme synthesis and secretion of some of the penicillin enzymes, some of the functional also happen in mesosomes, but the major function is helpful for the formation of septum during cell division, that is especially a binary fission. Here are the storage granules and inclusions. Here the storage granules act as a reserve food materials. So the amount of energy, whatever the bacteria can be required, the extra energy that can be stored in storage granules and inclusions, mainly functions as energy production. So here the bacteria can store this food material in the form of glycogen and fat droplets. Here the organ where the bacteria can store its food material is poly beta hydroxybutyrate. There will just it is very important. There is a chance to ask in exams. So what is the storage food material of fungus? That is poly beta hydroxybutyrate. PHB is the food storage organ of bacteria. So these granules are all those things will majorly act as the energy generating. Now we will see few of the inclusions that may be metachromatic granules are volatilin, polysaccharide granules, lipid inclusions, sulfur granules, magnetosomes, phosphorus reserves, energy reserves, and a few of the iron oxides, protein covered cells. These are all the few of the organelles which have a minor role in bacteria. So these are all the different types of cell structures or the components, functional components which are present in the bacteria. Coming to the last one, that is a specialized structure which is called as endospores. It's a very important. These endospores are especially produced by gram-positive bacteria only. Now we will see what is endospore. It is a thick-walled, asexual, heat-resistant resting spore. heat resistant resting spore which is produced by the bacterial species like Bacillus and Clostridium. So these endospores are produced by the bacterial species like Bacillum and Clostridium, which are comes under gram positive bacteria. So this endospore have a capability to survive even in 100 degrees, and few of the bacteria that Robert Koch or a few of the scientists worked in earlier days has a capability to survive even 120 degrees centigrade. So how capable it is, remember. So how, the scientists are getting to this, how these endospores are capable to survive even in extreme temperatures. Then they will come to know that these endospores contain an acid called dipicolinic acid. Dipicolinic acid. This acid is the mainly responsible to survive or thrive in extreme environmental conditions. So the dry weight of this endospores ranges from 5 to 15% of the bacteria. So due to the presence of an acid called dipiconic acid, it is helpful for the bacteria to survive in extreme environmental conditions. So whenever the favorable conditions will occur, what will happen? This endospore can germinate and it can produce a new bacterial type. The main function that protect our adverse environmental conditions, especially heat, highly resistant to heat, UV and toxins. So these are all the different types of functional organelles and all those things are which are present in the bacteria.